Today, I will be talking about a paper titled Single Cell Lineage Analysis Reveals Genetic and Epigenetic Interplay in Glioblastoma Drug Resistance. The bioinformatics method is sequence filtering. First, the introduction. The scientific question is, can certain genomic amplifications of loci in glioblastomas help find the cancer's resistance to drug therapies? The hypothesis is a combined lineage tracing and barcoding and single-cell RNA sequencing treated with receptor tyrosine kinase inhibitors can reveal the interplay of genetic and epigenetic mechanisms of drug resistance in longer-term sunitinib-resistant cell populations. Next, we have the key terms of the paper defined. 1. Single-cell RNA sequencing. This is a method of sequencing the same tissue type to find different functions and jobs of the different cells present. 2. Dasatinib-resistant cell populations. These are cancerous cells that have a resistance to tyrosine kinase-specific inhibitors. 3. Genetic and epigenetic mechanisms. These are acquired genetic events and persistent phenotypes. 4. Malignant cells. These are cancerous cells that grow and spread throughout the body by metastasis. 5. We have the receptor tyrosine kinase inhibitors. These block the tyrosine kinases, which are usually high in levels in cancers, to keep cells from growing. 6. We have GSC8 gliomospheres. This has the PDGFR amplification that allows it to adopt a drug-resistant phenotype to survive initial drug treatments. 7. We have the TSNE visualization. These reveal a strong coordinates between the two experiments that will be talked about later on. And this also reveals two main clusters of cells that are distinguishable by the expression of cell cycle genes. Next are the take-home findings. First, the scientists combined an lentiviral transgene barcoding system with a single-cell RNA sequence. Here, the DNA barcodes obtained would be transcribed and then captured in a single-cell RNA sequence. With the diverse barcodes being transcribed, rare subclone/tumor cells would be detected in the malignant cell populations in the DNA sequencing analysis. Next, the scientists performed two experiments. One experiment will be used to target DNA sequencing and assess barcode distributions. Then the same experiment was performed, but with a separate parental culture of GSC gliomospheres with cellular lineage barcodes for the second experiment. The first experiment had dominant jackpot lineages that made up 18.2 to 65.5% of the cell population replicants that represented more than 2,000-fold expansion of cells after just 70 days. The second experiment had all 6 replicates containing a jackpot lineage between 52.4 to 88.5% of the cell population replicates in just 56 days, which were already visible around 28 days. This meant that it must have an early advantage to resist the drug treatment. The second experiment was also dominated by the same lineage being the most abundant in the same barcode ID, E86 back. Experiment 2 displayed how genes in the E86 back lineage have many genes residing in a single chromosomal band, chromosome 13. The role of this chromosome's amplification in RTK inhibitor resistance have been reported in specific cancers. The transcriptional programs of these individual cells were analyzed by individual cells for the TSNE visualization. This suggested that cell populations from the same patient-derived gliomospheres are adapting to targeted RTK therapy. Experiment 1 replicatively displayed some degree of drug-resistant reversibility in clonal isolates with high copy number amplifications that had more stable dasatinib resistance. This concluded that non-jackpot genes lost their drug-tolerant phenotype when cultured in the absence of dasatinib, consistent with reversible genetic resistance mechanism. This concluded that combined lineage tracing and single-cell RNA sequence revealed that there was an interplay between complementary genetic and epigenetic mechanisms of resistance in heterogeneous glioblastoma. Why is sequence filtering important? First, it helps find the similar sequences that will be inserted in the database. You can also isolate certain parts of the sequences, and then they will look over the entire database for comparison. It will help find rare sequences that you might not have heard of, especially for cancers, and that is what they are doing in this paper. Next, Why is this important and what has been done before? In the paper, they talked about using a low coverage whole genome sequencing. This is like the next generation sequencing. This was used in clinical oncology to advance treatment of cancer. This was also used to identify rare cancer mutations by sequencing large number of genes in a single test. How are the results presented different? In the study, they focused on a single cell analysis rather than a large number of genes, like in the next generation sequencing. So a mixture of single cell RNA sequence and lineage tracing were used to find the interplay of genetic and epigenetics and the resistance of the tumor cells to the therapies. The study was able to isolate a specific chromosome that related to the drug resistance. What are the limits of this study's applicability? The limitations to the study was not only some of the reads were relevant to the analysis, means the experiment started to get lineage and chromosome specific. How was bioinformatics method used to support the results? To the left, you can see that figure 2 displays how single-cell analysis helped identify the chromosome amplifications and drug-resistant lineages. Before going to depth what figure 2 is, I want to ask, what is sequence filtering? Sequence filtering is when you isolate certain tissue cells and focus your sequence on that same tissue cell. Sequence filtering uses specific programs to be able to identify particular types of sequences or find rare types of sequences. This narrows down where the cancer cells are most abundant, such as in specific chromosomes/lineage. The scientists were able to find early cell growth replicates that led them to suggest that many cancer cells were found in a singular lineage that resided mostly in a chromosome. Here you can see figure 2A. Figure 2A showed the total number of single cells that were present in a certain number of lineage barcodes. And figure 2B showed the number of cells at day 28 of both experiments 1 and 2 with the TSNE visualization. You can see that in day 28, experiment 2 showed many more cells than experiment 1. Next, we have figure 2C. This showed the cell cycle signature scores with the TSNE of the experiments. Next, we have figure 2D. This showed the correlation of the jackpot lineage E6 that here shown in red and the other lineages in the TSNE plot. Figure 2E showed the fraction of cells that were positive in cell cycle signatures that were assigned a jackpot E86 back lineage and the other lineages. It is noticeable that the E86 back lineage had a much higher fraction of cycling cells, which means that it was present more than other lineages. In figure 2F, it showed the chromosomal bands that were most present in the E86 back lineage, the three significant bands of chromosome 13. These bands are mostly found in the E86 back lineage, so they were narrowed and isolated. Here we have an example of a BioPython sequence filtering code. The first line are the values of the sequence that are compiled in the list. Here you see the function that is used to narrow down your sequence or what you want removed from the sequence. If it reaches a certain number or fails to reach a certain number, it will be filtered out. The value inputted that return as true will be kept in the sequence. The value inputted that you return as false are the values that you do not want kept in your sequence. Next, we have evals. This allows you to create new Python integer objects. Here you filter the entire list of values along with constraints you made with values. With this code, you are printing out the list of filtered sequences. There are some questions I have about the code. One, is there any way this code can narrow down a certain sequence all the way down to a certain chromosome? Two, how can two different data be pre-processed in the sequence filtering all at once? Three, how big of a sequence can be allowed to be processed in this code? Is there a limit?